Okay, good morning, everyone. Let's get started. So today we're going to continue on. We're going to leave the world of molecular biology, and which, many of you might be happy about. Fungi don't really have any particularly exciting evolutionary milestones because they're just microscopic organisms. But as always, if you have any questions, you can drop them in the chat. You can ask them to me. Just as a final reminder, you do have your exam is due tonight at midnight. So if you have any questions, let me know sooner rather than later so I can actually answer the questions for you. But again, there's 45 multiple choice questions. It's all based upon material, not from this week, but from the previous 2 weeks. So, but yeah, so let's get this started. And so just before we sort of go on and talk about this, I wanted to talk about viruses a little bit. Just to talk about how we think viruses came to be, because we talked about how life evolved on this planet. And we talked about that mainly through a view of bacteria, and we also talked about a view through eukaryotes as well. But one of the sort of interesting things is we don't quite understand how viruses came to be. There's a pretty strong line of evidence of how microorganisms came to be and how we as eukaryotes came to be. But the question is sort of out there in the open is how did viruses come to be? And so there's sort of three hypotheses that we can actually examine for how viruses came to be. And so the first one is called the virus early hypothesis. And if you sort of remember back to our talk about RNA as a nucleic acid has the capacity to self-replicate. And RNA is something that can be synthesized in a primordial earth. Now, early virus hypothesis states that we had some RNA develop and it got encased inside of so one of those early membranes, one of those mycelia. And as over evolutionary time, it increased in complexity and essentially led to a virus, which is essentially, as we talked about, a parasitic organism. So that's one idea, that there was a pool of self-replicating RNA and it got encapsulated and eventually became parasitic. So that's the first hypothesis. There is some evidence for this, but we're not going to sort of dive into it. The second hypothesis is what we call the regression hypothesis. And this one has much more evidence for it relative to the other two. And the way this works is you have some sort of cell that is parasitic in nature. And this will come up later today when we sort of talk about the origins of eukaryotic cells. But the idea being you have some sort of cell that is parasitic, and it essentially undergoes what's called reductive evolution, meaning that it's losing parts of its genome, and it's transitioning from being essentially larger, being able to make its own energy and stuff like that, to being an obligate parasite, which is what a virus is. So it's something that is essentially undergoing reverse. You can think about it as reverse evolution, but it's really more of sort of it's taking a different track overall. And then finally, we have the escape gene hypothesis, which is there is essentially a piece of your genome or a bacteria's genome that had some sort of gene that was selfish. It had the capacity for self-replication, like we sort of talked about up here, and it eventually escaped from this. And because it escaped and it was self-replicating, and it's this idea that it was a selfish gene, it would try to propagate itself as a virus. And so this last one is the least sort of supported, but it is supported by the idea that every virus essentially has to attack a host, and it has to know the host very well. So being an escaped gene from a host makes sort of a little bit sense here. But as I mentioned, this regression hypothesis is the best supported line of evidence at the moment. But anyways, just wanted to touch upon that because we're talking about the origins of all sorts of life, and we don't really talk about how viruses came to be. And one of the reasons we don't do that is because viruses are incredibly hard to understand. I think if COVID has taught us anything about viral biology is that it is really hard to understand. We look at sort of COVID and we look at it relative to related diseases, and COVID is doing things that no one ever predicted a virus really could do until sort of COVID came around. So viruses are tricky, they're tiny, and they're kind of a pain in the butt. As a microbiologist, I can unequivocally tell you they are a pain in the butt. So but with that out of the way, let's go back to our clock. So we spent In terms of our sort of trek through this time period, we spent a lot of time talking about how the Earth was formed, how life came to be, what evidence there is for early life, and essentially how early bacteria arose, in particular how that great oxygenation event occurred. But now we're going to, just to sort of remind you where we went, remember the first bacteria out there were heterotrophic, meaning that they ate organic or carbon-based material. And then they were shortly there followed after by cyanobacteria, which are the very first synthetic, photosynthetic organisms. These cyanobacteria still exist today. They still provide you with about 50% of the oxygen you breathe. But these are just some microfossils of ancient cyanobacteria. As you can see, they're very, very small. But the cyanobacteria led to the oxygenation event of this planet, seeding the planet with oxygen. And as we mentioned, oxygenation happened really, really fast. And so you can just see this is time billions of years ago. And this is the atmospheric concentration as a percent of present day. This is a log scale. You don't need to understand scale, just understand the trend. No oxygen, no oxygen, no oxygen. And then bam, once cyanobacteria evolved, you get an explosion of oxygenation, and it happened within 50 million years or so, and it's called the Great Oxygenation Event, or some people call it the Oxygen Revolution. But this is an important event. Without oxygen, as we all know, oxygen is used in respiration. It's the way we get the way that most eukaryotes get most of their energies, by using oxygen in their respiration. So it was a really big thing. Because if we had no oxygen on this planet and everything was doing their metabolism in the absence of oxygen, life would stay microscopic. You need oxygen. You need it to get more energy out of food. Because if you ate a pound of sugar, again, it's a very important event here. One of the sort of the main things it does overall is it does provide an ozone layer. And so when biological life produces oxygen, that oxygen will react inside of the inside of our atmosphere, producing ozone, which, as we talked about, ozone being O3, essentially protects us from ultraviolet light. And as we talked about, ultraviolet light is particularly damaging to DNA. And if you're going to be living not in the water, you need to have some sort of overall protection. And the ozone layer is a great way to do that. So not only does oxygen allow you to get more energy from your food, but it also directly protects you if you're going to live on land. And sort of thus far, we've been talking about, we've talked about the finches and the Galapagos Islands, but that's in an evolutionary context. But thinking about how we've really talked about life that lives in water, right? We've been talking about microorganisms. We're going to talk about fungi, which typically also live in some sort of aqueous solution. But we're going to talk about plants later on today, and those clearly and definitively live on land, in addition to the many ones that live in the ocean. But when we think about plant, we think about something that lives on land. So I'm actually going to skip this because I don't really think it's that important. But just as a reminder, what the tree of life looks like, remember we have our bacteria, their cousin / our cousin, the archaea, both microscopic organisms denoted as prokaryotes. Remember, these are single cellular, they lack any meaningful organelles. And then we have our eukaryotes here. That's sort of the vast majority of things we think about when we physically can see them, because most of the eukaryotes that we know, at least have described, I should say, are large. Microscopic eukaryotic organisms are, but we have all these other groups of eukaryotes that we don't really see because they're microscopic. But in terms of what we know, again, remember, eukaryotes represent only a small portion of known life on this planet, and the vast majority of life exists here in these prokaryotic branches over here. But do remember that we all did at one point share a universal common ancestor that split evolutionarily, leading us to bacteria, archaea, and eventually eukaryotes. So today we're going to come back to this clock. We're going to talk about how we got our first multicellular organisms, how we got organisms with nucleus, and how we got plants and animals. And, sort of like thinking just sort of numbers again, we've been talking about a lot about this Archean eon here, um, very old fossils, oxygenation events, stuff like that. Now we're going to take a sort of a huge jump up in our history of time, basically 1 billion years, and start talking about eukaryotes, how they came to be, and then how we sort of shifted from eukaryotes to thinking about plants and thinking about animals, and so on and so forth. So. Sort of just remember there are two distinct groups of cells, prokaryotes and eukaryotes. Remember the key distinction between eukaryotes and prokaryotes is not only their size, but also the presence of these organelles. And these organelles just didn't sort of happen, right? Their ancestors had no organelles. So to make eukaryote, we need to think about how we physically got organelles, how we got all this stuff that we can encapsulate inside this much larger cell. So how do we get here? And we'll start with the nucleus. We'll think about how we got the nucleus, because the nucleus, as we've talked about, is the brain of your cell, contains basically all your eukaryotic DNA. Do remember that the mitochondria and the chloroplast have DNA, and we'll talk about those organelles separately in a minute because they are important milestones in sort of the evolutionary track of life. But remember, like every organelle inside your cell, it has its own membrane. Everything is membrane bound inside of a eukaryotic cell. So the question we should ask is, how do we get the nucleus? And so our best sort of evidence right now of how we got a nucleus is through what we call the syntropic theory. Now, the syntropic theory is simply illustrated in this cartoon thing that I made in PowerPoint here. It's very, very simple theory. The idea being that we had some sort of large archaea, and this large archaea engulfed this small bacteria. And I mean actually engulfed, like just ate it. Like, think about your white blood cells eating something inside you, right? That's what's happening. And this is a pretty common lifestyle in nature, something engulfing another organism to completely consume it. So predatory behavior is pretty common in the eukaryotic world. Now, sometimes when you engulf things, it might not be a bad thing. Maybe by engulfing this and you were going to eat it, well, maybe this organism provides you some sort of benefit, right? Maybe it provides you some sort of thing that's good, right? So think about if you adopt a dog, right? The dog is cool, right? You have this dog and he's a drain on your resources, but dogs are pretty awesome. You could take them for walks. They cuddle you, right? They play fetch. They have all these other benefits to you. So it behooves you to keep that dog around. And the same idea with this primitive cell. We have this large archaea consuming a small bacteria, and they form this symbiosis, this relationship where they start to depend on one another, start to give things to one another that they didn't have before. And so the idea being is that this small bacteria is providing something to this archaea. It was providing extra genes, potentially extra metabolic pathways, potentially other ways to combat other microbes, or with the capacity to eat new types of food. And so there's something happening here that we can physically look at and say, okay, well, maybe there's some benefit here. And this idea of in symbiosis, this sort of living together, will pop up again in a few slides. But remember, nothing lives by itself. And as we'll talk about with the human microbiome later in the class, very rarely do things sort of act on their own. Things that typically live together act in a consortium. And you can think about this like the human population, right? We like to think maybe we don't act on our own, but we really, as a society, act in consortium, right? Whether it's for or against one another. So bacteria and archaea are the same way. They live together, they act together, they do things together. And so it's not really out of the realm of possibility that things would, if something got engulfed by them, that they could potentially form some sort of symbiosis. And we've seen this sort of happening in real time. We've seen organisms evolve symbioses in the laboratory setting. But in addition, when we look at the membrane lipids inside of a modern day eukaryotic nucleus, they actually resemble the lipids of many groups of small bacteria. And so there is some biological evidence here, and as well as some laboratory-based evidence where we can actually show this happening in real time. So this is how we think the nucleus came to be. This idea of syntropy, where we have this idea of something engulfs something else, and they form this symbiosis or this relationship. Now, another idea for the nucleus is this is a mutation. And so there are a group of bacteria that live currently, and this is what they look like. They are what we call planktomycetes. They are very, very common in marine sediments and marine waters. And these bacteria are pretty cool. Sorry about that. And they have what we call a purulosome. And so the idea being is that they have this nucleoid region where they house their DNA, and then they have this purulosome. And this purulosome is a really interesting sort of nucleus-like structure that they essentially use to perform their really interesting metabolism. And this group of bacteria actually does a metabolism that's called anaerobic ammonia oxidation, or ANAMOX for short. And this bacteria is kind of cool because during this metabolic process, it actually produces hydrazine, which is actually rocket fuel. So it has this really interesting structure to house this really interesting metabolism, because it produces hydrazine, because hydrazine is a really, really toxic chemical. So it has this special compartment to keep this really hard to do metabolism in check so it doesn't spread around. Now, the idea being is if this mutation is if you evolve the structure, well, it kind of looks like a nucleus. It has similar proteins to a nucleus. Maybe this was the proto-nucleus. Maybe this is what early nucleuses look like. And as cells got bigger and bigger, it sort of transitioned to being more of a modern nucleus. But this does look a lot like a nucleus as is. A membrane-bound organelle with a nucleus, with a pile of DNA in the center. So this already starts to look like a nucleus, and maybe this is something that got more complex over time, leading to a eukaryotic nucleus. The only problem with this idea is that this is the only group of bacteria that we know about. And just to sort of go back to this tree here. Oops, one more. Our planktomycetes are over here. They're a more modern evolutionary lineage here. And so the idea being that they would cross over to form eukaryotes is not really supported by science, but it is a really interesting theory. Maybe there's something back here in time that looked like planktomycetes that then evolved, but as of now, there's not a ton of evidence that supports like maybe something like planktomycetes made eukaryotes. But it's an interesting sort of thought overall. And so that's the nucleus. And there's, as you see, there's lots of other types of organelles, right? We have our cytoskeleton, we have the Golgi, we have the endoplasmic reticulum. These are all, a lot of them are involved in transport and movement, or something like the lysosome, which is involved in the degradation of materials. But the key sort of ingredient that is really different outside of the nucleus from these types of cells is not really how things are moved around the cell, but really how we're getting their energy. And as we talked about, organisms need a mitochondria to get energy, and plants need a chlorophyll in the chloroplast. And so to explain this last sort of ingredient, how we got these energy producing or energy consuming organelles, we're going to turn to what's called the endosymbiotic theory. And this was proposed by a woman named Lynn Margulis in the 90s. And she's a really interesting woman. She's a professor emeritus at UMass Amherst, so I guess our sister school. And she's, as most women in science are that do really sort of high-profile work, she's pretty amazing. And if you have a few minutes, I would recommend taking a peek at her wiki profile. But she was pretty heavily ridiculed for this theory when it did come out. But now it's a pretty widely accepted theory as to how life sort of got more complex. So if you have a moment, read about a badass woman. But we're going to talk about what she proposed and what evidence exists, in particular modern evidence exists, to support this idea of the endosymbiotic theory. Now, the endosymbiotic theory is pretty straightforward. The idea is very much like that syntropic theory. We have something that was an ancestral prokaryote. It started to develop a nucleus. It started to develop primitive organelles like the endoplasmic reticulum or the Golgi apparatus. But again, we're trying to think about how we got mitochondria and how we got chloroplasts. But the idea being is it's very similar, endosymbiotic, meaning some larger cell engulfed a smaller cell, and there was a relationship evolved. There was a symbiosis evolved. And the idea being is that there was some ancestral heterotrophic bacteria that was engulfed by the sort of the last common ancestor between photosynthetic and non-photosynthetic organisms that led to the mitochondria. And for our photosynthetic organisms, after they evolved, or they consumed, I should say, the mitochondria, well, they engulfed a cyanobacteria, a single-cell cyanobacteria that then became chloroplasts. And that's the idea. Organisms living together to become much, much more complex. And this is something we see, again, across nature, right? We do it here in the human population. We live together. We do incredible things, right? But we talked about some symbioses earlier in class, right? Symbiosis between giant worms and bacteria at the bottom of the ocean, right? We talked about plants and microbes living in the soil together, right? Symbiosis is a really common thing in nature. And we're going to talk about more symbioses later in this class, but living together, working on one common thing together is pretty common in nature. And so this idea, this theory is, well, it's pretty common in what we see now, and it is supported by a sort of a long line of evidence that our mitochondria and that our prokaryotes are descended from free living bacteria that were engulfed by something bigger. And the evidence we can look at is first the membranes. And so many of our membranes, in particular the membranes of chloroplasts and mitochondria, essentially look identical to the membranes found in bacteria and archaea. So that's line of evidence one. Second, and it's sort of actually kind of an interesting one, is that many of our organelles are actually susceptible to antibiotics. And so the idea being is if you take an antibiotic, such as chloramphenicol, it can actually destroy some of the organelles at the cellular level. Just as a reminder, antibiotics only work on bacteria. So the idea being is that an antibiotic could kill an organelle, suggests that that organelle was potentially a bacteria in its past life. Because again, it's not toxic to us, it's toxic just to the organelles, which I think is cool. In addition, our organelles, in particular our mitochondria and chloroplasts, actually undergo their own reproduction. So during mitosis, your chloroplasts, if you have them, and your mitochondria actually independently replicate themselves. So they sort of do their own thing outside of mitosis, very much like a bacteria do, and they actually reproduce by binary fission. They don't actually do mitosis. These organelles reproduce themselves by binary fission. Our chloroplasts and mitochondria, as I've mentioned, have their own genomes, and these genomes are circular, round, and naked, exactly like our bacteria and archaeal genomes. And then finally, their ribosomes are almost identical to prokaryotic ribosomes. So there is a long line of evidence here that suggests that our organelles, in particular these mitochondria and these chloroplasts, originated directly from endosymbiosis of free-living cyanobacteria or a free-living heterotrophic bacteria. And I think it's cool. And the other thing I want to mention is that the size of a chloroplast and the size of a mitochondria are about 1 micron. And if you remember back to our microbe class, 1 micron is the average size of a bacteria. So they look like them, they behave like them, they have similar genomes to them, and it's a really sort of strong line of evidence that suggests that endosymbiosis is the primary mechanism as to how we got our mitochondria and our chloroplasts. Again, we don't have chloroplasts, but plants got their chloroplasts. So really, really strong evidence. But I just want to sort of show you some really cool data, because I'm one of the things I like about microbiology is sort of all this evolutionary history stuff about microbiology. But one of the things that's kind of cool is you can look at the DNA inside of mitochondria and compare it to bacteria. And the idea being is that every eukaryote on the planet has a mitochondria, right? If you are a eukaryote, you have a mitochondria. It doesn't matter if you're a plant, doesn't matter if you're amoeba, doesn't matter if you're a blue whale, right? You have a mitochondria. So what this really points to is that there was one event way back in evolutionary time, a billion years ago, half billion years ago, whatever time frame we want to give it, that there was some endosymbiosis event that led to all eukaryotes having mitochondria. So there's essentially one event here, one endosymbiosis event here. Now, when we start to look at the DNA of our mitochondria, we can actually look at it how it compares to bacteria. And the group of bacteria that we actually see our mitochondria sort of grouping with in an evolutionary context is actually a group of bacteria called the alpha proteobacteria. And the alpha proteobacteria are a really important group of bacteria. You know, they're really important in agriculture. They do all sorts of different stuff. They're not particularly pathogenic in terms of what they do, but they are very, very common across the planet. So for instance, this bacteria here, Pelagibacter, accounts for 25% of all cells on the planet. So, you know, I told you there's 10 to the 30 bacteria on the planet. 25% of those are just Pelagibacter. So really, really common group of bacteria. But when we start to look at the DNA analysis, we start to see, well, evolutionary speaking, our mitochondria fall out in this group of bacteria called the Rickettsiales, which points to, again, one origin, but it also is sort of an interesting spot where this falls out. So it falls out in the Rickettsiales, and some of you may have heard of Rickettsia before, this genus of bacteria here. Some of you might have heard of Wolbachia, or maybe you probably never heard of any of these other ones, but you might have heard of Rickettsiales or rickets. And what's interesting about this group of bacteria that our mitochondria are evolutionarily related to is they're actually endoparasites. And so one of the best known diseases that we know is caused by rickets, like a Rickettsiales bacteria, is Rocky Mountain spotted fever. So this is caused by a bacteria called Rickettsia rickettsii. It is an alpha proteobacteria. So again, it falls in this tree here. It's right here. It's very closely related DNA-wise to mitochondria. But it is an intercellular parasite, and it's something that's actually propagated by ticks as well as woodland creatures. So this is an image of a cell. You can see the nucleus here, but you can see this bacteria that's an intercellular parasite of this organism. And if you want to learn more about Rocky Mountain spotted fever, go to the CDC. Always great information at the CDC. But the idea being is that, well, mitochondria were likely some sort of intercellular parasite that essentially invaded a host a long time ago and essentially just stayed. It didn't cause any diseases. It just sort of lived in a symbiosis. So I think this is a really fascinating sort of avenue here, that you had this parasite, it infected something, and instead of sort of killing it, it just sort of lived in harmony with it. And eventually, through evolutionary time, they just got propagated together. And again, it all points to one origin, all within this alpha proteobacteria group, again, which this group here is all intercellular parasites. If you want to read about another cool intercellular parasite, I'd recommend looking up this bacteria here called Wolbachia. It is an absolutely fascinating organism. They're parasites of insects and crabs and stuff like that, and they do all sorts of wacky and crazy things like manipulating the sex of the organisms and the biology. They do all sorts of cool stuff. But that's our mitochondria. Again, likely one origin, because all life that is eukaryotic has our mitochondria. It is something every eukaryote needs. And again, it's likely arose through some sort of thing that resembles an intercellular parasite. And the idea being is you can actually look at the life cycle of Rickettsia. It invades the cell. Once it gets inside the cell, it lives there. And essentially escapes it, killing the cell. And so this is the sort of life cycle it lives. But you can imagine, like, maybe if it invaded a cell and the cell starts to deal with it like an infection, well, maybe it just sort of stayed inside that membrane and just lived there because it was a good place to live. So that's the sort of the overall idea of Rickettsia and the sort of origin of mitochondria being related to something like Rickettsia. So chloroplasts, they are different. Than our mitochondria. They, again, only plants have them, or only things that are photosynthetic. So it's a very small number of the lineages of eukaryotes have them. But when we look at the DNA of our chloroplasts, there's actually seems to be multiple symbiosis events across all the different lineages that we see here. So these are all the different lineages of photosynthetic organisms. So we have the very old ancestor, the cyanobacteria, and then we have all these series of different types of plants. So we have land plants, algae. We have all these different types of, like rhodophyta is red algae. This is single cellular algae, and there's all these different types of algae and microscopic photosynthetic organisms. But what you'll notice is that on this tree, we have multiple different circles. And what these circles tell us is that there's actually what we think to be multiple independent symbiosis events. So the idea being is that even though cyanobacteria evolved the capacity to photosynthesize a long time ago, the capacity for bigger things to photosynthesize evolved multiple times. So there was multiple endosymbiosis events by different groups of microscopic bacteria that happened throughout this tree. And one of the sort of the interesting things you might notice is that this tree is colored in different colors, and these different lineages actually use different types of chlorophyll. And so we talked about chlorophyll as just sort of this generic light capturing molecule in our third class together, but there are many different types of chlorophylls. And so this is what chlorophyll looks like. Chlorophyll A and B are the most common types of chlorophyll you see in land plants. But there is all sorts of other ones. There's C, C1, C2, D, and F. So this is what eukaryotic chlorophyll looks like. It's this big ring structure of carbon, and it often has this tail. And these different structures of chlorophylls do correspond to these different colors here. Their different lineages have different chlorophylls, which have different structures, which capture different wavelengths of light. So remember, when light is coming down from the sun, essentially it has all sorts of different wavelengths as a whole. And these chlorophylls have evolved different capacities to capture different wavelengths of that light. Now, if you look at chlorophyll in bacteria, which is called bacteriochlorophyll, you can see that all the chlorophyll are very similar to the bacteria, right? They have that same ring structure. All bacterial chlorophyll have this little tail, but you can see some eukaryotic chlorophyll has actually lost that tail. So there is some evolutionary thing going here, but it all, again, stems from what bacterial chlorophyll look like. So the idea being is that our chlorophyll are very, very similar to bacterial chlorophyll. And actually, it's actually kind of interesting. If you pull the chlorophyll out of a regular plant and stick bacterial chlorophyll in there, you can actually use the bacterial chlorophyll. So it still works. So there is that evolution relationship. But remember, very much like our mitochondria, there seems to be an endosymbiosis event. And again, it's all by looking at shared structures, shared genomes, shared ways of replicating. And in the case of our photosynthetic organisms, very similar types of chlorophyll overall. Very small evolutionary changes between the two of these. So this is sort of how we got our organelles. It seems to be a case of endosymbiosis, bacteria evolving something a long time ago, and something bigger taking advantage of it. And sort of the big two broad classifications of eukaryotic cells, again, are plant and animal cells. But sort of how those are divided up on the tree of life sort of varies. So we think about plant cells, they're really only found here in plants. Everything else is sort of a classical animal cell in terms of this basic structure here. But now we're going to sort of move on, think a little bit harder about plants and fungi. And we're talking about plants and fungi because historically speaking, they sort of came first. In particular, the fungi really came first, it seems. Well, something that resemble fungi seemed to come first. And now we talk about plants and fungi together, and it's actually more of not really of a evolutionary relationship because fungi are more closely related to us than they are to plants. But historically speaking, plants and fungi were just actually dumped into the same group. Plants and fungi historically were treated as the same thing, but now we know that plants and fungi are completely different. And so when you go out to the forest and you see a mushroom, that's a fungi. It's not a plant, it's a fungi. But let's talk about these organisms. Let's talk about what they're good for, what sort of diversity there is. What are some of the major milestones of evolution of plants and fungi? What are some of the major groups of these two organisms? So we're going to start with the fungi. These are microscopic organisms, primarily speaking, and so they fall into three broad classifications of fungi. They have the yeasts, which are single-cellular microscopic organisms. They look very much like bacteria. These are the groups of organisms that produce your beer, your wine, and your bread. Most tasty things are made by yeast. We also have our molds, and so these ones you all are very familiar with. If you leave your bread out for too long, or if you leave any food out for too long, you'll see a hairy thing growing on your thing. Well, that's because you're seeing molds growing on there. Microscopic organisms that are piling on top of one another, giving you that physical pile of mold that you see on your bread or whatever you left out. And then finally, we have our mushroom-producing fungi, and these, again, are microscopic organisms that are producing these very, very large structures. In the case of mushrooms, some mushrooms can be huge. They can be feet across. They're actually kind of crazy sized. But producing these bodies by piling and piling and piling lots of microscopic organisms on top of one another. But mushrooms do arise from fungi living inside the soil that are, in fact, microscopic. Just as a note, because we're not going to talk about the biology of mushrooms, mushrooms are simply just reproductive organisms. So these fungi produce mushrooms to release spores to then go fertilize essentially the gametes of another fungi, just as a note. So, but what are our fungi good for? What roles do they play in nature? What roles do they play in your life? Because you might not sort of think about mushrooms or fungi in any other way besides food, but these organisms do play a critical role in nature as a whole. So their primary role in nature is in the soil and as well as on rotting wood. And these are as decomposers. And so they break down dead and decaying things, and they recycle all that energy and all that carbon and all that nitrogen, all that phosphorus back into a biological system. So they are really important recyclers in nature. And this is one of the main things that they do. So for instance, when a tree dies, it breaks down not because of the bacteria, it breaks down because of the fungi. And fungi are eukaryotes, so they're much bigger than bacteria, so they can break down a lot more material than a bacteria could. So fungi are huge decomposers in the soil. There are millions of them per gram of soil, so they are very important for decomposition. Fungi also produce some very important symbioses, including lichen and mycorrhizae, which we're going to talk about in depth because they are pretty cool symbioses, but they're also really important for nature as well as humans. They are useful as food, so they can be farmed. We think about them as mushrooms. And then finally, they are parasitic and pathogenic. So they have a number of roles that they play. But the first type we're going to talk about is mycorrhizal fungi. And this is a symbiosis between a plant root and a fungus. And so this is a microscope view of a plant root colonized by fungus. And this is a type of fungus called ectomycorrhizae. And this type of fungi, what it does, it just essentially grabs onto the outer root surface of a plant and basically just attach on there and live there indefinitely, essentially until the plant dies. So that's ectomycorrhizae. And then we have what are called endomycorrhizae, which physically take their structures called hyphae. They basically think about them as roots, and they essentially just pierce the plant root. So they actually just physically live inside the plant while also sticking outside the plant root. Now, what these fungi do is not harm the plant. They actually form this really important symbiosis. And so what they essentially do is act as a way for a to physically protect the plant. But the other thing that they do is they actually act as a way for plants to get more nutrients. So these fungi and the fungi are helping the plants get bigger. And now you might not think that this is an important thing, but pretty much every major agricultural crop that we have, corn, wheat, soy, so on and so forth, all form mycorrhizal associations. Pretty much every plant that we can think of forms one of these associations, whether it's endo or ectomycorrhizal. So they are super duper important for plants. Life on this planet, in terms of plants, would be way less effective if it wasn't for these fungi. So remember, bacteria produce half the oxygen you breathe. The other half of the oxygen you breathe comes from plants. And plants could not produce as much oxygen as they did if it was not for the mycorrhizae on these on the roots. But more importantly, agriculturally speaking, we couldn't produce as much plant biomass for consumption without these mycorrhizae. No matter how much fertilizer we dump on our plants, they won't produce enough biomass for us to eat if we didn't physically have these fungi on the roots. So really, really important symbiosis for plants, both in an agricultural setting as well as a natural setting. Very, very cool symbiosis. Very, very heavily studied symbiosis because, again, super duper important. Next up is a sort of an overlooked type of symbiosis, and this is the lichen. And you all have seen these. You probably just discounted them. You probably never really noticed them, maybe. You see them everywhere. You see them on trees, as you can see here. You'll see them on cement. You'll see them on buildings. You'll see them everywhere. You see them on rocks. And these lichen, they're pretty unremarkable from basically what we normally see. And one of the sort of interesting things is this is what we think about when we think about lichen, but in natural settings, so if you go deep in the woods away from humans, you start to see very beautiful types of lichen, like blues and oranges and reds and pinks and stuff like that. They take all sorts of cool shapes like this or this. And sort of one of the actually interesting things is that these lichen as a whole are actually pretty sensitive to pollution. So this type of lichen you'll only find in very pristine areas, whereas this type of lichen, here you see that everywhere. It handles pollution very well. But just to know, like, even though this is what you see, like basic gross greens and maybe some blues and some blacks, as lichen sort of everywhere, even around the campus at UMass, lichen can take some really beautiful shapes and colors. But this system is a symbiosis between an algae, which provides a house for a fungus, and bacteria. So basically this algae is a symbiosis of, this algae is sort of the main body you see here, and inside it's housing a fungi and a bacteria. Well, sometimes multiple fungi and multiple bacteria, but for simplicity's sake, we'll just say one fungi and one bacteria. The idea being is that, well, this algae photosynthesizes, and the fungi and the bacteria provide this organism with different types of metabolites, things that it needs, like amino acids and nucleotides and things like that. So they sort of act together. They sort of feed each other in sort of this space that they're living in. So they live together, but again, it's all inside the algae. Sort of one of the interesting things about lichen is they cover about 6% of the Earth's surface. And now we think about the surface of the earth being like the ground, or being rocks, or being streets, right? But surfaces is everything, right? It's plants, it's rocks, it's buildings, it's sidewalks, it's, a fence, right? That sort of encompasses surface, right? It's not just natural things, or it's not just the ground. Surface is really essentially anything, right? You could technically think of like a tree trunk or a rock. That's all surface, right? And so lichen are really abundant. They've colonized 6% of all the Earth's surface. Again, 6% of everything that could potentially be colonized. That's a lot of surface area. 6% of the surface area is a lot of area. Think about how big the planet is. It's like basically like colonizing the surface of Mercury. It's huge. It's a massive, massive area. One of the other things that's interesting about lichen, they are very, very long-lived. So if you see a big lichen like this on a rock, chances are it's hundreds and hundreds of years old, because to grow from something very small to very large takes a very, very long time. They're very, very slow growers. Another thing that these organisms do is they contribute to weathering. So if you have a lichen on a tree, or if you have a lichen on a rock surface, they'll actually contribute to the breaking down of that rock through the production of chemicals during their sort of normal biological activities. So they help weather rocks, help weather surfaces, things like that. And then finally, one of the most important things these lichen do is they're actually pioneer species. And so what I mean by pioneer species, they colonize bare areas. So you can imagine, like, if you're out on Hawaii and there's a volcanic eruption, and you have all this brand new clean magma that's solidified, forming this nice solid rock. Well, you look at that solid rock and there's no dirt there. There's nothing living there. And that's how all the Hawaiian Islands were formed, right? They're all this just plain bare area that was eventually built up to look at this amazing lush jungles that are on Hawaii, right? Well, to get there, it required lichen to colonize. So the idea being is you have what are called pioneer species, and lichen are the very first pioneer species. They land on an area, they colonize bare space, they bring nutrients to a place, and once they're colonized, once they start bringing these things start to accumulate dirt and debris. You start building soil. And as you start to build soil, you can start bringing in more complex things like grasses and small shrubs and weeds. And then you start bringing in these bigger and bigger and bigger things. You start to build up more and more soil. You start to build up the system. So you can go from a bare rock, like you see down here in this diagram, to lichen, to bigger things and bigger and bigger and bigger, leading you to very, very lush things. You give it a, I guess, a pat on the back, because without lichen, there'd be no beautiful islands like Hawaii. More importantly, early colonization of land was important by lichen as well. So, in addition, we have been using our fungi as food, and so fungi are rich in carbohydrates and proteins. Outside of insects, they are one of the most nutritious things you can eat, pound for pound, because of how rich they are in carbs and protein. And we do grow them in industrial capacities. They aren't photosynthetic, remember, so you grow them in basically sheds. They grow on dirt. It's really, really simple. Sometimes you also grow them in bags, like you have these oyster mushrooms here that are growing in bags of dirt. So they are really pretty commonly eaten. Pretty much every culture eats some form of mushroom. But we have been growing them. They're a really good source of food. Again, lots of protein, lots of carbohydrates. These organisms can also be pathogenic. They can make you very, very sick. They also have been known to be parasitic. And so one of the sort of things I fear most as a human being is getting a fungal infection. And one of the reasons is because fungal infections are really hard to get rid of. We think of bacterial infections or even viral infections being hard to get rid of. Fungal infections are even harder to get rid of. So there are numbers of pathogenic fungi out there, including Cryptococcus or Aspergillus or Histoplasmosis or Candida, that have some sort of environmental reservoir that can then infect the host, whether it's your lungs, your mouth, the vaginal cavity, your intestine. There's all sorts of places fungi can infect on your body. There are also some groups of parasitic fungi out there as well, but they are pathogenic. They're not as common as bacterial pathogens because they are much larger than your bacteria, but they are pathogenic, and they are really, really good pathogens. And one of the nastier pathogens that we're finding out is this bacteria called Candida. So for those of you that have ever heard of this concept of a yeast infection, whether it's on a male or a female, it's all caused by Candida. And this Candida it lives naturally on your body. It lives naturally in the vagina. It lives naturally in a number of different places, but it is the most common source of yeast infections on both, again, males and females. Because of it's so common, we're actually seeing the rise of Candida as a serious growing threat. So we talked about how antibiotic resistance is a problem, right? Antibiotics being compounds that treat bacterial infections. Well, there's an entire other class of drugs called antifungals, as you would imagine, treat fungal diseases. And so we've been using these to treat Candida infections. And as you would imagine, as you keep using something clinically, well, we're starting to see evolution of resistance against these antifungals. And so, in fact, there is a superbug called Candida auris that is resistant to basically every known antifungal out there and can kill you in a pretty rapid pace, actually. It's kind of a nasty fungus, but very much like bacteria, our fungi can evolve the capacity to be resistant to against medications that are designed to kill them, and they can be deadly. And so we're actually starting to see, at least before COVID, we don't really hear about it very much because COVID sort of takes over things, but there was this huge rise in Candida auris infections that were resistant to multiple antifungals that was spreading through the globe. So it's a really nasty group of organisms. So And the other sort of the last thing I wanted to mention about fungi is overall they are an important group. They decomposers, they them in that capacity, but they do form this huge backbone to how things work naturally, both in terms of pathogenic organisms, but also just how nature works as a whole. So fungi are this sort of underappreciated group of organisms as a whole. And the other sort of last thing I wanted to mention is if you do like mushrooms, just remember that some of the deadliest toxins we know do come from fungi. So if you ever like, I'm gonna eat this mushroom I found in nature, don't do it unless you 100% know what it is, because again, very deadly toxins produced by fungi. And in terms of an evolutionary perspective, toxins are being produced so they don't get eaten, right? You evolve a defense to prevent you from getting eaten, right? So that's our fungi. Again, they're more closely related to the animals, but historically speaking, they were grouped with the plants. And plants are sort of one of the more important things for humans, right? They produce a ton of oxygen. They provide materials for building for us, right? They give us food and sustenance, right? They do a lot for us. And so next up, we're going to talk about the plants. And when we're thinking about an evolutionary context of plants, very much like early microbial life, the earliest plants would have been water-bound. And you're thinking about seaweed and algae, right? Things, if you've ever been to the beach and you see a pile of seaweed, you see a pile of algae, that's what early plant life would have looked like. And it wasn't till the development of the ozone layer that plants had the potential to move onto land. But for something to move from from the ocean to the land, it's a lot of difficulties. And the question you could ask yourself is, what difficulties, if you were a fish or if you were a plant living in the ocean, what difficulties would you face if you were moving onto land? And in particular, what's on land that's not in water? And the two key things that we have to think about here are gravity. Gravity is not really a thing in the ocean. I mean, it is, but you have water holding you up and sort of mitigating the effects of gravity, right? So gravity is a big deal on land. In the water, you have the water to sort of hold you up, right? To keep your body afloat or to keep your body from collapsing in on itself. You don't have that on land. If you don't have the structures to hold yourself upright, gravity will crush you like a pancake. That's first. And then the second is water. When you move from water to land, you're no longer continuously surrounded by water. And as we talked about, all life needs water. So if you're going to move onto land from the sea or from the lake, you need to be able to find a way to capture water to keep yourself wet, because everything needs to be wet. And again, the two main challenges here are gravity, because gravity is tough, right? You have to have the structures to hold yourself up. So early plants, when they moved onto land, they would have been very, very small, very, very low-lying. Think about like very, very tiny, tiny shrubs, or very, very tiny blades of grass. Like that's what we're talking about. And not the sort of huge trees you're thinking about that are 50, 100, 300 feet tall. Early plants would have been tiny, very, very tiny. And again, the second problem would have been desiccation, drying out. So desiccation just means drying out. And early plants would have had to figure a way to not dry out, because again, everything needs water. And you don't have to worry about drying out when you live in water. So to go from water to land, you have to figure out how to get over these main two struggles, drying out and gravity. Just as a sort of note, like if you guys ever thought about a struggle that you faced, did you ever think like gravity was a struggle? Probably not, but early life would have been a struggle. Drying out, definitely. Everyone's thought about dehydration at some point in their life, but definitely not gravity. So how do all these things affect our plants? What did the evolutionary tract of a plant look like? Well, there are four major groups of plants. I'm sure you're not going to recognize some of the plants in these groups, but that's okay. They're not that common. But the four major groups are our non-vascular plants, our vascular seedless plants, and then our gymnosperms and our angiosperms. And so most things that you know of fall into this latter group, the seed-producing and flower-producing groups, these gymnosperms and the angiosperms. Most of the things we eat, again, fall into this group of plants here. But historically, evolutionary speaking, they had to evolve from something. We had to go from simple plant to slightly more complex plant to very complex plant. Because you might not think about it, but an apple tree is really, really complex. And that's in contrast to something like a fern, which is not very complex. So we had to work our way up the evolutionary tract. And we're going to talk about some of the major sort of life cycle changes that had to happen, the structures that needed to evolve, that let us go from simple to much, much more complex. But again, it started with some sort of ancestral prokaryote, protist here, so a microscopic eukaryote that engulfed some sort of cyanobacteria to get those chloroplasts. Because the key characteristic that all these plants share is they're photosynthetic. They produce their own energy. And it doesn't matter if you're a cool apple tree or you're something sad like a fern. Well, I don't think ferns are sad, but relative to an apple tree, a fern is certainly sad. You have to photosynthesize. So it all started with some sort of endosymbiosis, whether it was, some ancestor or something more closely related to each of them, but some sort of endosymbiosis had to occur to give them chloroplasts. And in terms of a time period, the origins of plants is about 500 million years ago. So relatively speaking, we're talking about a really modern time. 500 million years ago to the present is a really modern time, right? We're talking about dinosaurs, coming to be around 200 to 150 million years ago. So plants evolved not that far in the past. I know 500 million years is a long time, but in terms of the planet, remember that's 1/8 of the history of the planet. But there was some, we have these green algae. These are essentially water living plants. There was some origin of plants that occurred, again, 400-ish million years ago that led to the divergence of all four of the major lineages. And the most modern sort of lineage of plants, again, are these angiosperms. These are the flowering plants. And these happened about a smack dab in the middle of the reign of the dinosaurs. So. But you'll notice that most of evolutionary time was sort of, plants that don't really sort of fit the mold as to what we think about for most plants as a whole. So let's start off with the very basic. Let's start off with the non-vascular plants. And these include our mosses, our liverworts, and our hornworts. And you probably have only seen mosses. We don't really see very many liverworts or hornworts here in Massachusetts. But they do exist in Massachusetts. They're rare. But you see them elsewhere in much more pristine areas of the country. And these are all non-vascular plants. And what do I mean by non-vascular? Well, vascular, think about it like your vascular system. Some sort of tissue or some sort of channel inside of a plant to transport nutrients from one area of the plant to another. So they had no way to pump nutrients from one side of the plant to the other. Now, the question you should ask is, how do they move things around their cells, right? You look at this moss right here, right? And this rock is big, right? So how does this moss move things centimeters, right? How does it move things an inch? Well, the answer to that should be in your brain, and that answer should be diffusion. And all non-vascular plants rely on diffusion and osmosis to move water and to move nutrients around their cells. And so they have no way to actively move things. They just sort of let things flow in between them. And now that's how water-based plants do things. Our algae all rely on diffusion. They have no vascular tissue. So very much like our biophytes, our non-vascular plants, they relied on diffusion. They relied on osmosis to move things around their cells. In addition, these organisms reproduce by producing spores. So they produce gametes that they just release out. And they rely actually on water to distribute their gametes, their sex cells, their spores, to fertilize another plant. And they also have what we call a haploid and a diploid life cycle, meaning part of their life they live with half the number of chromosomes, and the other half of their life they live with a full set of chromosomes. So let's look at what this looks like. So the idea being, and we start off here, we have a haploid moss. So a moss, pretty much its entire life lives with only half the number of chromosomes that it potentially has. Now, once this is haploid, it's going to start to produce, it's going to think about essentially wanting to reproduce. So the idea being is, as I mentioned, they release spores. They release spores, which you would think of as like eggs. I'm sorry, like sperm, and they rely on rain or flooding events to essentially release their spores to carry them to another moss. And so once that happens, it gets carried to another moss. You can imagine this isn't a very effective way to make sure you reproduce, because you're just relying on, you know, random rain events to do this. But it gets carried to another moss where that spore, again, that sperm, fertilizes an egg, forming an embryo. And once you form that embryo, well, now you're diploid because you have a fertilized egg. Then you start to produce spores, which then go out and form new mosses. And those new mosses are going to be haploid. And the whole process repeats over and over again. So they have this period of being haploid, but when they get fertilized, they start to produce spores, which then go out and seed new mosses. They become diploid. So this, again, it's all reliant on water to transport these things for fertilization to happen. So it's a very, very simple life cycle. This is how algae do it in the ocean as well. So this is very, very early plant life, relying on diffusion and osmosis, and relying on water to transport your spores, to transport your sperm and your egg to make the next generation, to reproduce. In terms of mosses and stuff like that, we've been using non-vascular plants such as moss economically as well as agriculturally for a long time. So people will use them to essentially, for cooking. So historically speaking, we've been using mosses for tinder sources to cook food and stuff like that. We've also been using them in gardens as like a fertilizer. You grind them up; they're great fertilizer. We also use them as flood control. So you can build structures out of moss. They're very strong. They're very sturdy. They last a long time to help with flood control. So mosses might not, this might not seem like very much like cool stuff, like, an apple tree or something like that, but they do have some uses overall to the human population. But again, no vascular tissue, very simple life cycle, completely relying on osmosis and diffusion to move stuff around the cells, and relying on water to move their spores, to fertilize their eggs and stuff like that. So next up, we've evolved the capacity for vascular tissue. And so this leads us to our vascular but seedless plants. And these include ferns and horsetails. And you all have probably seen ferns around. You may not have seen a horsetail, but they do exist here in Massachusetts. And there are also wacky and wild giant ferns out there that exist in the tropics. But when we think about what these are, they have a vascular system. Again, think of a vascular system like your bloodstream. They have this specialized tissue to transport things over longer distances. Now, what you're seeing here is you're going from teeny tiny plants that are very, very low to the ground to much bigger things. Ferns here in Massachusetts can grow upwards of 3 feet tall. Horsetails can grow 4 or 5 feet tall. The name of this plant is escaping me right this second, but it can grow upwards of 10 to 15 feet tall. And so with the evolution of a vascular system, vascular tissue essentially allows you to grow larger, to actively move, say, water or nutrients from the soil up into your leaves, or vice versa. And so this is a big sort of it's pretty straightforward. We think of trees as just being like sort of hardwood, but inside of the outside of the bark, you have all this nice soft tissue that is the vascular tissue. In particular, you have essentially two tube systems. One's called the phloem and the other one's called the xylem, that actively transport things from the top of the tree to the bottom of the tree and vice versa. So phloem brings things from the canopy down to the roots, and the xylem brings things from the roots up to the canopy of the tree or the crown of the tree. This is true for trees, this is true for ferns, this is true for horsetails. Anything that has a vascular system has xylem and it has phloem. And just as a sort of a reminder back from our very first class about the chemistry of life, all of this is not based on diffusion, but it's instead of the cohesion of water, right? Water's ability to adhere or co-adhere to itself allows water to move up and down these tree trunks, allows it to move through the phloem and through the xylem from the roots to the shoots, or the shoots back down to the roots. So this important property of water makes vascular tissue actually work. If water wasn't cohesive, this vascular tissue wouldn't actually work. You need something like a heart to physically pump it up and down the tree. But because water has this cohesive ability, it can move things up and down the tree. And just to sort of show you, what this, if you want to see like a cartoon diagram, this we have, our phloem. It's sort of interesting. Typically speaking, when we move stuff up from the roots and up to the shoots, it's mostly water. But it can be other things, but it's primarily water and nitrogen. But when we're moving stuff down from the canopy to, say, the roots, it's oftentimes sugar, in particular the products of photosynthesis. And so phloem often has, often has these little sieve tubes to help filter out stuff that is not, they don't want down to the roots. And it also, on the way down, the phloem will deposit or pick up things from leaves on the way down. So, but again, xylem goes up and phloem goes down. But again, very simpler tubes that are reliant completely on water co-adhering to itself and then it adhering to the plant wall to move things up and move things down. Now, going down is also reliant on gravity, just as a note. But this is xylem and phloem, and if you wanted to look at what xylem and phloem looks like, this is what xylem and phloem looks like at the microscopic level. It looks like something screaming inside of a plant tissue, but you can see all the plant cells here, and this is what xylem and phloem look like. Xylem and phloem. And when you're eating something like celery, or when you're eating something like anything that's crunchy and wet, you're eating vascular tissue. So when you eat celery, you're eating all this crazy face of vascular tissue. And I found this little cartoon last year that I thought was pretty funny, because this is what xylem and phloem, this is what vascular tissues look like. And they look, again, it looks like someone's screaming. And I thought this was a pretty funny cartoon. So I thought I would share it with you all. Maybe this will make you pay attention, because you think maybe there's more cartoons coming. There's not, but there might be. So, but if we're going to talk about the life cycle of our vascular but our vascular but non-seeding plants, we'll talk about the life cycle of a fern. And ferns are pretty straightforward. The idea being is that you have an adult fern. It produces these haploid spores. So remember, having half the amount of the chromosomes, but adult ferns are diploid, unlike our mosses, which their adult stage is haploid. And these essentially accumulate on the back side of a fern. So if you go in September, usually, here in Massachusetts, if you start to look at ferns, you'll start to see these piles of spores on the underside of ferns here in Massachusetts. But the idea being is it produces all these spores, which again are haploid. It relies on the wind to carry them to a new location. So it's not relying on water like our non-vascular plants were. These spores land on the soil, growing in and forming into what's called a prothalsis, or a haploid fern. Then these plants rely on rain, allowing sperm. You can think of the male part of the fern to essentially swim from the reproductive structures on these plants to the female reproductive structures, thus fertilizing their egg. So they essentially fertilize themselves. So they essentially reproduce asexually. They can reproduce sexually, but most commonly they reproduce asexually. Again, their own sperm fertilizing their own egg. Then once that happens, you have an embryo, which then develops into the full-fledged fern that we see, if you go out into any forest. So, and that whole cycle repeats. But you'll notice is that adult ferns, unlike adult mosses, are diploid, but both these plants thus far have that, again, that diploid and that haploid stage. But again, we're getting slightly more complex. We're not relying on the water to distribute our spores. We're relying on the wind now. So this is something that you would only find on land, right? You can't have wind underwater, right? You can have water currents, but you can't have wind. Right? And again, we're seeing sexual reproduction going on here. So these are sexual organisms. Next up, and definitely moving into the much more exciting group of organisms, and this is what we call the gymnosperms. And these are conifers, cycads, gnetophytes, and ginkgos. And just as a sort of kind of a cool note, ginkgo, which is down here at the bottom, there's only one species of ginkgo on this planet. It's called Ginkgo bilboa, or Balboa, depending on how you want to pronounce it. It's the only ginkgo that we know of. And if you walk around the city of Boston, you actually see ginkgo trees around Boston. And the reason you do this is because these trees are really good at surviving in pollution, and they're really good at pulling pollution out of the air and stuffing it down into the ground. So these trees you'll see all over major cities, including Boston, because they are super duper hardy relative to pollution. But that being said, they are the only species of ginkgo on this planet. So they're the only living ancestor of whatever evolved to make ginkgos, just as a note. But when we're thinking about gymnosperms, we have our conifers. So these are pine trees, also redwoods. So these are things that typically produce cones. And commonly they make leaves that are essentially shaped like needles. So you think of like a pine needle. Well, that's a conifer. In addition, there's also what are called cycads and gnetophytes. These are tropical plants. You don't see these here in Massachusetts. You'll see cycads in Florida and throughout the Caribbean and stuff like that. Gnetophytes you typically will only see in very dry areas. I won't claim to be an expert on gnetophytes, so I'm not going to try to say cool things about them because they're not a very sexy-looking plant, to say the least. The rest of them are all cool and awesome, but gnetophytes, not very cool looking. But when we're thinking about our gymnosperms, they typically have needle-like leaves outside of ginkgo and these gnetophytes, and they all make cones or some sort of capsules for their seeds. And this is sort of the first plant, at least, essentially the first plant to evolve the capacity for meaningful sexual reproduction. And I mean sexual reproduction as in like one plant, like one tree will fertilize another tree completely, as opposed to relying on water to potentially bring you to a new thing, but most likely resulting in asexual reproduction. So the gymnosperms are really thinking about actual active sexual reproduction, and they actually physically can't self-fertilize themselves. So there's no asexual reproduction inside of the gymnosperms. We had a question. So a sago palm, they are gnetophytes. No, gnetophytes are these sad-looking things here. Most palms are. This is a specific type of palm, but they are gymnosperms. And so, as I mentioned, the major milestone of our gymnosperms here are seeds. This is a really important evolutionary thing. We don't think about it being like a big deal, right? Because we eat seeds all the time, right? Peanut butter, or we eat chia seeds, or any number of seeds out there, or legumes, right? We eat seeds all the time, but seeds was a huge evolutionary milestone in the world of plants. And what this does is allows plants to spread further, and it also allows plants to store things in the seed, in the soil, in what's called the seed bank. Now, it allows plants to spread things farther because it's not relying solely on the wind to carry things. What seeds allow you to do is allow you to have things eat them and take them far away from you. So they're not just dropping down, or they're not just potentially being carried 10 or 20 feet by the wind. You're actually, by producing a seed, you're actively relying on an animal to take your seed and disperse it hundreds of feet, or sometimes miles away, or maybe if your bird's hundreds of miles away, right? So seeds were important for this aspect. The other way seeds are important, well, actually there's two other ways. Seeds also are the result of fertilization, but they also have a huge amount of nutrients for early plants. Because you can imagine if you're if you're say a baby plant, you land on a if you're a seed and you land on soil, well, maybe that soil doesn't have all the nutrients that a baby plant needs. However, a seed does. A seed provides all the fuel, all the carbohydrates, all the nutrients that a plant needs to start early growth, to become a bigger plant, all packaged inside that seed capsule. So it's a really important thing. It allows you to drop pretty much anywhere and start growing, even if the conditions aren't ideal for a very, very small plant. So that's a really important thing about seeds. And then the final thing that's important for seeds is they are very hardy. So most seeds are pretty hard. It takes a lot to break them down. So if you've ever eaten chia seeds, and if you ever looked at your poop after you've done your business, you'll see chia seeds out the back end. Seeds are very hardy. They can hang around for a long time. They can pass through digestive tracts very, very easily. The other thing seeds can do is hang around in soil for a very, very long time. And when you have a bunch of seeds sort of hanging around in the soil for a very long time, this forms what we call the seed bank. And so picture this scenario. You have a forest. And in that forest, you have these beautiful trees growing, all these plants growing. It's a great place. But you have a huge fire. And the fire comes through and burns the entire forest to the ground. No more plants. So how are we going to get new plants into this area? Well, we have the seed bank. If you burn a forest down, there's tons and tons of seeds in the soil. So once the seeds can sense that, hey, stuff's getting better up there, the seeds start to come alive. They start to germinate. They start to form trees. They start to form new plants. They start to bring the forest back to life. So the seed bank is an incredibly important thing. So for those of you that followed what happened in Australia this past year and late last year as well, with all those monstrous fires fueled by really dry tinder from decades of climate change, what we're seeing here is these forests, these deserts are coming back to life because of the seed bank. The seeds that are in there are germinating, they're blossoming, they're coming back to life, bringing life back to these systems. So seeds are an important way, not only for plants to spread, but also for ecosystems to pop back up following natural disasters, or sometimes man-made disasters. But as you all know, seeds have an amazing variety in appearance and shapes, and they also have a huge variety of taste. You could think of something like beans, right? If you go buy some black beans, right? Black beans, they're seeds, and they look one way, they taste one way. But then you can go buy a pomegranate, right? And pomegranates are sweet, and they taste really good, right? Seeds have a huge variety of shapes, appearances, and desires from different organisms. So some organisms will eat different seeds relative to others, and they have different nutritional values as well. So seeds have, once they evolved, they sort of went down a crazy evolutionary tract. You can actually, just as a note, you can look at even seeds within closely related organisms, and they're really, really different. So for instance, we think of beans being sort of, like maybe black or brown or red, or maybe white, and they have that sort of distinct bean shape. Well, that's just not true. Beans have a huge variety of colors, of shapes, and mismatches of colors. Beans are crazy. They're all the same, same group of organisms, but seed evolution has gone crazy in the bean family, just as a note. One of the other things that our gymnosperms have evolved are these cones. And so for those of you that know what a conifer is, thinking about palm trees, you're thinking about redwoods, things like that. There are cones, and cones are ways for organisms to A, reproduce, and B, spread their seed around. And so the idea being is, if we think about a pine cone, there are male cones out there, and they look like this. You might maybe never have noticed them on a tree, but they are male pine cones. They're much smaller, and these male pine cones will actually release pollen that are carried by the wind that hopefully get taken to a female pine cone. And if this pollen releases, that these trees release and makes it to a female pine cone, it will fertilize this female pine cone, and then this female pine cone will then go on to produce seed, which would be pine nuts, which, if you like pesto, is delicious, right? And so this is how things go. Now, for those of you that have allergies like me, you are allergic to what gymnosperms and angiosperms have evolved, this pollen. as opposed to producing spores, they're producing pollen. And this pollen is much more akin to sperm than it is to a spore. So if you have allergies like me, you are allergic to tree sperm. And I know that sounds gross, but that's just the way it is. But ultimately, when we have a male cone fertilizing a female cone, this female cone will start to develop seeds, and eventually it will drop off in the hopes that something will come along, break apart this cone, eat the seeds, and take the seeds somewhere else, poop them out, and then a new pine tree or a new redwood can sprout up from where those seeds were pooped out. And that's the flow of information we're working with here. Now, when we're thinking about the overall life cycle of a gymnosperm, it's mostly diploid. So again, it has the full number of chromosomes. But the idea being is you have male cones and female cones. And the male cones are releasing the pollen, which again is the sperm, which tries to find its way to the egg or the ovule. And again, when it's a gamete, a pollen or an egg, or a sperm and an egg, it's a haploid. But once fertilization occurs, you produce an embryo, and that embryo then develops into a seed, and then that seed can then be planted in the ground and grow into a tree. What you notice is the vast majority of the life cycle here is diploid, which is in contrast to the previous three, which was half diploid, half haploid. So getting more complex here, the evolution of pollen, the evolution of seeds, and the evolution of these cones, this much more directed form of sexual reproduction. Now, I just want to make one last mention of this. Pollen from one gymnosperm here cannot fertilize the egg over here. You have to go to a completely new tree to do it. So this doesn't work. This requires direct reproduction, whereas the previous two groups of plants can fertilize themselves, but not a gymnosperm, and certainly not the next group of plants we're going to talk about, which are the angiosperms. And sort of just as one last thing, because class is just about over, I want to just mention one last thing about our gymnosperms, is they can get incredibly, incredibly old. Some of the oldest organisms on this planet and some of the largest organisms on this planet are gymnosperms. So I'm sure you guys have heard of those giant sequoia trees or the redwood trees up in California and Oregon that can get upwards of 300 feet tall, and you can drive a car through them. They're so massive. Those are gymnosperms. But some of the oldest organisms on this planet are actually conifers, these gymnosperms. And there's a tree that we know of out there that are over 5,000 years old. So they were around long before the Bible was written, long before the Torah was written. They were around like when humans were like discovering agriculture and stuff like that. So there are trees that are incredibly, incredibly old. And those are all, again, gymnosperms, cone-producing, seed-producing things. But from an evolutionary perspective, we added a lot of important things with this organisms. We added better vascular system, which we didn't really talk about, which allows you to pump things up from pump things very, very long distances, allowing things to get taller. The other thing we evolved is a structural compound called I'm sorry, I just had a mental fart about the structural compound. Lignin, there we go. We evolved a structural compound that we didn't talk about called lignin. That's very, very strong, that allows trees to make wood and get very, very tall. And we also evolved cones. We've also evolved this much more stringent form of sexual reproduction. We also evolved the capacity for pollen. And most importantly, we've evolved seeds. And seeds are important, again, naturally speaking. They're also incredibly, incredibly important sources of nutrients for billions of people on this planet. So without the evolution of these things, without the evolution of gymnosperms, there's going to be a lot of things missing from this planet. And ultimately, what evolves from the gymnosperms, which we'll finish in talking about next class, is the angiosperms. And these are the flowering plants. This is what we think about typically when we think about plants. This is what we think about when we think about crops. So next class, we'll talk about the angiosperms, and then we'll follow the angiosperms by talking about how animals evolved, how we got some of the major structures in animals that we see today. So with that, I will end class. If you guys have any questions, as always, I will hang around. Otherwise, I hope you guys have a great